So today we are joined by Jenny Tung, who is an associate professor in the Department of Evolutionary Anthropology at Duke University. Professor Tung is an internationally recognized leader in her field, carrying out fundamental research, examining the relationships between genetics, behavior, and life history in primates, especially baboons. So today, Jenny's talk is titled The Social Genome and Primate Evolution. So thank you for joining us today, Jenny, and welcome to Beck.
Hey, thanks very much, Brian, and thanks to all of you for being willing to carve out some time in your day to stare at a computer for another meeting. I'm really sorry that I wasn't able to be out there in person, but I guess we're all sorry that we're in the middle of another wave. So I hope I'll get the chance to catch up with some of you in person down the road. Okay, can you guys see my screen okay? This looks nothing embarrassing popping up my computer?
Looks great.
Okay, great. I'll go ahead and start by introducing the baboons, the study system that I'll spend most of my time talking about today. These are the two animals in my title slide, two females, Rwanda, and her then adolescent daughter, Rodeo. Rwanda is a particularly nice poster child, I guess, for a population because she has been the beneficiary of a particularly charmed life as far as being a baboon in the Amboseli region of Kenya goes. She spent most of her life as the alpha female or the second ranking female in her social group, and she's been quite reproductively successful. She's produced 8 live births, 2 miscarriages, and is still going. She had her last live birth just in 2020. This is probably in part a product of the social advantage she enjoys. High-ranking females in Amboseli tend to reach maturation earlier in this population, and they also enjoy shorter interbirth intervals. So they've managed to turn it around again, start cycling, and have another offspring pretty soon. Because a number of her kids are females, she's likely also to be surrounded in her social group with a number of close social partners who will remain with her throughout her life. And we found through almost a decade of work now that an individual female's ability to be integrated versus isolated in her group or to have relatively strong social bonds versus weak social bonds is one of the best predictors of how long she's likely to live. So in this graph from Archie et al. 2014, the blue line shows the life expectancy for the top quartile of most socially integrated females versus the bottom quartile of most socially isolated females. And you can see that the difference on average reaches actually a couple of years in magnitude, at least the amount of time it takes to produce another live offspring. Rodeo, by being born to a high-ranking socially integrated mom, probably also has enjoyed a number of benefits. Those are 2 major sources of early life adversity and advantage that we've explored in our population, which again has perhaps even more striking predictive effects on lifespan. So here we just counted up the number of good things and bad things that can happen to baby baboon females in our study population to find that those who were born in a silver spoon sort of lifestyle with no major sources of disadvantage tended to live on average about a decade longer than those who experienced 3 or more major sources of disadvantage. It turns out that these major predictors of life course outcomes, social status, social integration versus isolation, early life social adversity versus disadvantage, are things that appear to be fairly common predictors of outcomes in social mammals like the baboons that we study and like our own species. A couple of years ago, some colleagues and I tried to pull together what information had been published to date on natural lifespan in wild social mammals. And we found that in general, there are very consistent relationships between these sources of social advantage and adversity and overall life expectancy. So here I'm showing you some of the aggregated results at the time for different measures of social isolation, social support, social integration, and so on. And by and large, with perhaps interesting example of the yellow-bellied marmot, and I know Dan Blumstein couldn't be here today, but he could tell us more about that, certainly. The pattern seems to be that in social mammals, in unmanipulated natural populations, those individuals who are more integrated, who have stronger sources of social support or more stable social bonds, live longer. This is actually by far best studied in our own species, where where the relationship between social integration and social support and natural all-cause mortality has been described for over 100 years. What I'm showing you here is an attempt to aggregate much of that data together that was published in a meta-analysis about 10 years ago by Julianne Holt-Lundstad and colleagues. This is about 150 studies of adult humans of both sexes across a number of different countries, but concentrated in relatively wealthy developed nations. What it shows is the relative evidence for an effect of social relationships measured by a social support or social integration on mortality, a predictive relationship, compared to some other major environmental predictors of life outcomes that you may be aware of. The further the bar is to the right, the larger the meta-analytic estimate of effect size. So what this is telling us is that individuals who have low social support or relatively low social integration face an increased predicted risk of all-cause mortality, excluding suicide, that exceeds that associated with moderate levels of smoking, past history of cardiovascular disease, abstinence versus alcoholism. Not shown here are obesity as a risk factor and exercise, which actually are smaller than the ones that I'm showing you here. And it's these kinds of results over time across lots of different population contexts that have led social scientists to call social factors like socioeconomic status and social support, quote unquote, fundamental causes of disease. Now, as evolutionary biologists, we may be less interested in disease per se and more interested in how health and or mortality risk translate into lifetime reproductive success, into Darwinian fitness. And we have evidence too in the baboons that anything that predicts lifespan is also likely to be a major predictor of fitness. So here's the relationship between age at death for female baboons in this Ambacelli population and the number of surviving offspring they produce over their life course. And this is for complete lifespans. Essentially, it's almost clock-like. A female in Ambacelli produces another live offspring about every 2.1 years. So if you think back to the sort of effect sizes I described to you earlier, a difference in 2 to 3 years of lifespan, that's the top quartile versus bottom quartile of social integration, may translate into one additional offspring, a fairly big effect in these slow life history species. A difference of 10 years for adult female survivorship is quite a serious hit to fitness. So these differences in the social environment not only affect the lives of individual baboons, but also their evolutionary legacy in subsequent generations in a fairly marked manner. So that leads us to the two questions that I'll try and talk to you about today related to the work ongoing in my lab. The first is a question about how this works in the first place, how social interactions might lead to trait differences that we believe are important to fitness on the background of a genome that doesn't change throughout the life course. And I'll be talking specifically about our work looking at changes in gene regulation as a potential connecting mechanism. And in the second part of my talk, I'll discuss a little bit about what we're thinking about how these sort of social processes influence evolutionary and population genetic change for a population or species as a whole, in this case, looking through the lens of hybridization and admixture. Okay. So I'll start by mentioning some work that my lab has done on Rhesus macaques, another highly social, highly hierarchical primate species, using captive manipulations that allow us to have some degree of experimental control over the social environment. Here we've asked a basic question, which is whether differences in social status alone, in the absence of other major differences in group demography or access to resources, can by itself influence different types of physiological and molecular outcomes. The reason we think we're able to do that is because in adult female Rhesus macaques, we can manipulate order of introduction into brand new social groups. And it turns out that individuals who are introduced earlier into a new social group where they don't know any of the other individuals in their new group become the highest ranking individuals in their group on average. And as you insert females after that point, they kind of insert into the dominance hierarchy directly below those individuals who came before them. So we can do that. We make these little groups of 5 females each. They very rapidly form a clear dominance rank hierarchy, as that's a sort of baked into the system of female Rhesus macaque sociality. And then we can replicate those groups. And we usually do that about 9 or 10 times. So we're studying about 45 to 50 individuals at any given time. To really nail down causality, we can watch them for, say, about a year or so, and then rearrange those social groups again so that females who are alpha in those initial groups, the top-ranking females in each group, get placed into a group together. All the betas get placed into a group together, and so on down the line. They again quickly reestablish a hierarchy, and that allows us to watch the same females when they're both high status and low status, or low status and transition to high status, or some number of them stay at about the same level in the hierarchy. And again, we can watch them in these sort of long-term stable hierarchies for a period of time afterwards to look at the outcomes. I won't go into the results from that work. That's been about 10 years of work with my colleagues Mark Wilson and Bas Mikopoulos at Emory University and the Yerkes National Primate Research Center, and Luis Barrero at the University of Chicago. But I will tell you the basic take-homes, which is that when we do these types of manipulations, we find that the social environment, mediated through differences in social status, which are largely assigned through randomization by us, results in changed patterns of gene activity, altered gene regulation for hundreds to thousands of genes. And many of the most pronounced effects we see of social status actually occur when the cells from those animals are challenged by other types of cellular stressors, like signals that simulate pathogen infection. Further, we find that at least some subset of these effects seem to be biologically embedded into the memory of the cell, so that if we sample individuals after that second manipulation, we see strong effects of their social status at the time of sampling, but we also see these kind of hangover effects of their social status over a year beforehand, even though past and current social status are by design completely uncorrelated with one another. So that's been a very interesting set of observations for us, but a natural question that might arise for many of you is whether the types of experiments we're doing in captivity in these very small groups of Rhesus macaques relative to normal group size for this species in a fairly artificial environment actually carry over to animals living in nature. And so this is where I'm going to turn back, and I'll stay for the rest of my talk, focused on the baboons of the Amboseli ecosystem in southern Kenya, which I'm lucky enough to study as part of the Amboseli Baboon Research Project, founded by Jean Altman and Stuart Altman in 1971. We're now, this is my first talk of 2022, so this is the project's 51st year of data collection we just hit now. I get to do this work because I'm part of a really wonderful collaboration between Jean, who's an emerita professor at Princeton now, Susan Alberts, who's here at Duke with me, and Beth Archie, who's a professor of biological sciences at the University of Notre Dame. We co-direct the project together, and it would not be possible without the contributions of all four of us, I think, in the form it's in now. And we also are able to do this work only because of the hard work and professionalism and talent of our long-term Kenyan field team. A subset of them are shown here. I just want to point out this smiling guy in the middle, that's Raphael Mutatua, who's been working with the project now since 1981. So many of our folks have been with us for a long time and are really extraordinary at their jobs and are responsible for the day-to-day monitoring that goes into all the data I'm going to talk about next. Okay, so what we mean by 1971 to present is that time span is the time span during which observations have been continuous, meaning we've been watching individually recognized animals in our social groups and their descendants. Those generations now are hitting the 9th generation for some of the families. Since that time, we collect daily demographic data, information on their ecological circumstances, what they're eating, who individuals are having both affiliative and competitive interactions with. And increasingly over the last 20 years, we've done more and more biological sample collection. including the type of sample collection I'll talk to you about here. Most of our work is entirely non-invasive, but there are times in which we want to be able to study something going on in the blood samples of these individuals, or to be able to collect morphometric data that we can't do simply by watching the animals. So occasionally we engage in these periodic darting efforts. And this is a picture of our very talented field assistant, Kinyowari Terry, who's been with the project now since 1995, in the process of one of those efforts. Essentially, we go out very early in the morning. We hang out and look as innocent as possible. That's actually Kinyua up here on the top, trying to look innocent. He's actually holding the blowgun, which is about a meter long metal tube, in his right hand. And we wait for an opportunity when the air is still, when there is no one in the group looking, and we deliver an anesthetic containing dart via this sort of handheld blowgun just through breath propulsion. That allows us to collect blood samples, which is what we're doing here, and to do the types of experiments, or at least a form of the types of experiments that we first pioneered in captivity. So this is work that was started by my former PhD student, Amanda Lea, who's now an assistant professor at Vanderbilt and transitioned to studying humans. And it's been carried on by my current graduate student, Jordan Anderson. And basically what Amanda said is, well, these sorts of cell challenges that you're doing with the macaques, I'm pretty sure we can do those in Ambacelli too, even though, you know, we're off the grid and we don't have a classical CO2 incubator and lots of other things that we had to adjust to. Nevertheless, I'm glad she pushed me to do that, because I think turns out she was right. Basically, what we do is draw blood into two pre-prepared tubes. One contains just cell culture media, so the basic ingredients that cells need to live outside the body, at least for a number of hours. And we draw at the same time from the same blood draw samples into a tube that contain cell culture media plus something else. For example, we often use lipopolysaccharide, which is a pathogen-associated molecular pattern that tells cells in baboons and in us that they're being infected by a gram-negative bacteria. We incubate those samples in parallel at body temperature. This is one of my favorite pieces of field equipment. This is a modified igloo incubator that's maintaining our samples at 37 degrees. And then after a set period of time, we harvest the cells and prepare them for downstream genomic analysis. And what I'll tell you about next is one type of analysis that we do very often, which is to measure genome-wide gene expression using RNA-seq, high-throughput sequencing-based techniques. So what we were interested in the work I'll tell you about today is the relative impact and differences in the signature of dominance rank, social status at the time of sampling, measured for both males and females in our sample, and a measure of social bond strength, which we call the dyadic sociality index, and has shown up in some previous work in Ambacelli. This is the per year strength of a female's social bonds to her top 3 closest social partners. And I just want to point out that although rank and social bond strength, or rank and other measures of social integration, are often correlated, these two particular measures in our population are not well correlated, at least for female relationships to other females. And for the dyadic sociality index, sorry, I should have mentioned, that will be focused on females alone. Okay, so the first thing that probably made me happier than maybe anybody else, but I'll show you anyway, is when we compared the results for dominance rank associated genes in female baboons in the wild in Ambacelli in Kenya to dominance rank associated genes in female Rhesus macaques, a different species, in captivity outside Georgia, what we get is a relatively strong agreement. What I'm showing you here are plots that group genes into distinct functions or pathways. And the particular pathways or functions I'm showing you here are cases that are strongly enriched in one direction or other for rank-associated genes. So for example, things on the positive end of the x-axis show pathways or gene sets that are enriched for high expression, more activity in high status females. And on the left side are gene sets that are enriched for higher expression in low status females. And in general, there's lots of stuff going on in this first quadrant and in this third quadrant, which are the two quadrants that agree in direction between what's going on between these two very different systems. There are a couple that disagree, which I think are interesting in and of their own right, but we were pleased to see that despite the substantial differences between these systems, the type of signature that we see of dominance rank on gene regulation in immune cells tends to be pretty conserved. Here I'm showing you that same kind of pattern, but I'm focusing specifically on all of the genes annotated in this molecular signatures database that we're using here in the inflammatory response pathway. So this is the effect size of dominance rank on gene expression for every gene in that pathway for baboons on the x-axis versus macaques on the y-axis. And you'll see that the vast majority of genes in this pathway are consistent with higher expression of inflammatory related genes in low status females in both of those contexts. One of the interesting things to us, though, is that when we looked outside of simply social status, when we looked at affiliative relationships as well as dominance relationships in the baboons, we actually see that they both have detectable effects, but they don't look the same. A lot of the stuff that's been reported by us and others about elevated activity in inflammatory and innate immune defense pathways, here these pathways shown in red, are more highly expressed in low-ranking individuals and have a substantial signature of social status, but nothing in particular out of the ordinary for social bond strength, at least at baseline. So in other words, for these key immune pathways, we see a strong bias towards a positive beta, a positive effect, so higher expression with low rank for female rank, but absolutely 50-50 direction of effect sizes and nothing particularly strong for social bonds. However, those distinct signatures actually change in nature if we look not only at the baseline samples, what's going on just when baboons are kind of walking around with nothing in particular happening to their cells, but when we look at the LPS challenge cells, the cells in a state that feel like they are being attacked by bacteria. So as I showed you earlier, we have two samples for every individual. So here's the gene expression of a particular gene. This happens to be a Krebs cycle gene in baseline in yellow versus in the LPS stimulated state in green. And you can see, for example, here that strongly socially bonded females, those are the ones to the right-hand side, have very, very pronounced responses to the experience of infection. Whereas relatively socially isolated females, or females with weak social bonds, I should say, respond much less to the experience of a simulated infection. And in fact, what we see overall is that if we change our phenotype to actually asking about the response to infection as opposed to just sort of gene expression within a given condition, we see almost no effect of dominance rank on the response to infection, versus we see about 200 genes in which with the response to infection, they actually within individual magnitude of that response is substantially affected by the strength of social bonds. So one interesting question would be to ask, well, since the signatures of social status and the signatures of social bond strength are not exactly the same, right, are in many ways different from one another, what happens to individuals who are doubly socially disadvantaged or socially advantaged? So here I'm showing you the median gene expression, just as a summary of overall expression of all genes annotated in the inflammatory response. On the left-hand side are those weak social bond females. On the right-hand side are those strong social bond females, where the colored box plots differentiate females who are lower than median rank versus females than higher than median in rank. So you can see that rank effect, that main effect of rank here, but you can kind of see perhaps why the social bond effect is not particularly noticeable in a main effect analysis. But here, when we look at these females in particular, these are individuals who both have strong social bonds and are living at the top of the dominance rank hierarchies in their group, or towards the top of the dominance rank hierarchies in their group. And they look distinct in having systematically lower expression of inflammatory related genes in their cells. This is another way of looking at the same kind of result. Here's genes involved in the inflammatory response, the median inflammatory response expression on the right-hand side. On the left-hand side, another innate immune pathway involved in inflammation and antiviral signaling. And then each line here is a different female in our sample, again, just stratified by whether they have stronger than median or less strong than median social bonds, or higher or lower status than the median. And again, here what we see is that these individuals down here are the ones who have seemed to have systematically different and lower levels of expression of these genes. They're the ones who are both doubly socially advantaged and walking around with a basic immune profile that differs from the other females around them. Neither Rodeo nor Rwanda are in this particular sample, but I'll just point out that a number of their female family members, Rafiki, Reese, and Rhoda, actually are, and they too tend to be high-ranking, relatively advantaged individuals in their social groups. One of the other interesting things we were able to do, which we couldn't do in the Rhesus macaque sample, was to contrast what the signature of social status looks like for female baboons who attain and maintain rank through this nepotistic process, where females tend to insert under the hierarchy in the dominance rank hierarchy directly under their mothers, and male baboons who have an entirely different way of determining and defending their social status. They do so by a direct physical competition with other males. So here, I'm going to show you a scatter plot where every dot is a different gene, and I'm plotting the effect size of dominance rank on gene expression in males against that same effect size for the same gene in females. And what we find is a striking correlation. But it's a striking negative correlation, meaning that genes that are more highly expressed in low status males tend to be characteristic of high status females, and vice versa. And just as there's a positive correlation between the effects of rank in captive macaques and wild baboons, there's a negative correlation between the male baboon effect sizes and the effect sizes in female macaques. So in other words, baboon males and baboon females look directionally opposite to each other in terms of what social status appears to be doing to the regulation of their genes in these circulating immune cells. Another way to look at that same point is to ask about whether we can use gene expression by itself to predict the dominance rank of an individual who donated a sample. And when we do that using elastic net regression, just a basic machine learning technique for fitting a predictive model from high dimensional data, we find that if we use male data to predict male dominance rank, or female data to predict female dominance rank, we get a reasonably good positive correlation between predicted rank and expected rank. But if we use the male model and then ask it to predict rank for females, or the female model and ask it to predict rank for males, we get this negative correlation. In other words, based on gene expression alone, high-ranking females look like low-ranking males, and low-ranking females look like high-ranking males. And that particularly tends to apply to genes that are involved in those basic immune defense processes, like the inflammatory response and so on. So based on our work in Amboseli, we can add to some of the things that we think we've learned from the Rhesus macaques by saying that the social environment is also a primary predictor of gene regulation in immune cells sampled in completely unmanipulated wild populations. But how that signature manifests depends on the aspect of the social environment we happen to be testing. So I think tend, or at least we in the Amboseli project tend to come to our work thinking that, well, high status is preferable to low status for these animals. A high degree of social integration or strong social bonds are preferable to weak social bonds, but they don't necessarily do the same thing when we look at individual biological outcomes. And that seems to match with some of the results that I'll show you in a little bit on life course outcomes like survival too. And for the dominance rank differences between males and females, we think this is mostly explained, or our hypothesis is that those differences are explained by paying attention to the actual dynamics that shape the particular environment of interest. We use the terms dominance rank, we use the term social status to apply to both males and females, and there's a sense in which they are parallel, because they describe the ability of a particular individual to exclude individuals of the same sex from resources of interest to that sex. However, the consequences at a physiological and molecular level, we think are probably really closely tied to what you have to do to be high or low status. And again, for males, to drive it home with this picture that Beth took of two males having a conversation about their relative rank, this is the way that males attain high rank. And this is the way that they have to sustain it by regularly winning physical competitions with other males. And females don't do it in the same kind of way. This may be physiologically costly to them, but it's worth pointing out something that I think probably you all already know, which is that attaining high rank, at least in a system like this, is pretty much the only way you're likely to have success from a Darwinian standpoint. So this is from one of Susan Alberts' papers in 2006, showing the relationship between male dominance rank. One is high, and then it goes down from there, and the number of offspring produced per opportunity to mate with a conceptive female. Right. So something that is beneficial from a fitness standpoint may be costly from a physiological standpoint. Alternatively, it may be beneficial to have an upregulated inflammatory response at a point in time when this is the type of behavior that you're engaging in. This hypothesis seems to be supported by some other recent work that we've done, led by my graduate student, Jordan again, and a former postdoc of mine, Rachel Johnston, who's now at the Broad Institute, looking at one measure of biological aging in the baboons we study. That is to use an epigenetic clock, measures of DNA methylation across the genome that are used to predict age actually fairly accurately. This was, of course, pioneered by one of your colleagues at UCLA, Steve Horvath, who has shown that that epigenetic clocks can fairly accurately predict chronological age and also biological aging on top of that in many, many different organisms. These are the results of a clock that we calibrated for the Ambicelli baboons from whole genome bisulfite sequencing data. It's not perfect, but it's pretty good. And we were interested in what kinds of environmental or social experiences explain the deviation from perfect prediction. That is, what explains why a particular individual baboon looks older when we measure its DNA methylation levels than predicted for its age? What explains why other individuals tend to look younger than their actual age based on our demographic data? We asked this about measures of social bond strength, and we asked this about measures of early life adversity, because both of those things predict lifespan in Ambicelli. But it turns out that the only thing that we were able to robustly detect in our analysis was, again, an effect of male dominance rank. So here's again dominance rank on the x-axis, high ranking, high status individual on the left, going to low ranking on the right. And this is the deviation from true chronological age on the y-axis. High ranking males look systematically old for age, whereas relatively low ranking individuals look systematically young for age. And it turns out when individuals change their dominance rank within our sample, when we have longitudinal samples, their epigenetic age also tracks where they currently are sampled. So we think that this is starting to get put together a picture of the differences between the impacts of these disparate sources of social advantage and adversity. And it was really nice that some of Susan Alberts' work with two junior scientists, Pancho Villavicencio and Fernando Campos, happened to be coming out around the same time, where they too showed with, with actual lifespan as an outcome, which is really kind of where the rubber hits the road, right? That females and males, that social bond strength and dominance rank have different effects on survival as well within females, similar to what we're observing at the molecular level, where they don't have shared effects, where their signatures are distinct. And that female and male social status have sex-specific effects on survival. Social status is not a major predictor of lifespan in the female baboons and Ambicelli, whereas in males there's a hint of an effect, but that effect goes in the direction where high status males have somewhat higher levels of mortality risk at any given time. Okay. So that picture is still evolving, but we think it's becoming both increasingly rich and increasingly complex as we ask about the various pathways that link the social environment on one hand to different types of outcomes on the other. So now I will switch sort of scales of inquiry for the rest of the talk to talk about not only how social processes influence what's going on very immediately in the body, but how those processes might end up influencing evolutionary genetic change over the long term, specifically the types and nature of genetic variation found within wild primate populations. And one of the best places to ask this question is in the context of admixture and hybridization, which lead to fairly large-scale changes in the pool of genetic variation available within a population to work in. And while natural hybridization has been well studied for quite some time in wild primates, it's also become increasingly of interest in understanding and contextualizing the history of our own lineage. As you all are probably well aware, our conceptualization of human evolution has been really revolutionized over the last 10 years or so by ancient DNA analysis, which show that rather than sort of simple bifurcating trees leading to the ancestors of modern humans, we are part of a much bushier set of events that include interchange between divergent lineages of archaic hominins over quite some period of time. This is from some coverage of work by Svante Pääbo and his colleagues on introgression and hybridization in humans. It's from 2018, so of course it's terribly out of date now, but you can already see that there's a lot of patterns of gene flow that are connecting what used to be considered fairly discrete lineages. Of course, one limitation in our own lineage is that we are the only species of Homo left, and so it makes it very difficult to understand or infer what's happening in those early generations post-contact. That's something we can do by studying primates in natural hybrid zones. So Ambocelli is located kind of right here in the center of this yellow box on the in the area where the ranges of Anubis or olive baboons meet the range of yellow baboons, shown here in red. In the 1960s, when Jean and Stuart Altman scoped out Ambocelli as a potential place to study baboons, they wrote that it was clear that the animals in the Ambocelli area were yellow baboons, or Popiocynocephalus. Fast forward a couple of decades, and in 1982, Amy Samuels and Jean Altman recorded the first observation of a Papio anubis, an olive or anubis baboon male, into Ambocelli, followed by integration into the social groups and subsequent breeding events with that male and immigrants who came into the population further on. So we have a hybrid population starting in the early 1980s. For those of you who don't study baboons, this is what I'm talking about. This is an Anubis baboon taken near Lake Nakuru, picture taken near Lake Nakuru in Kenya, versus a kind of a poor picture of a yellow baboon. It's poor because I took it in the Tsavo region of Kenya to the east of Ambocelli. This is a yellow-like Ambocelli male, and this is a hybrid male in Ambocelli. Given what we know about this male's pedigree, he's actually a back cross he was an F1 hybrid back cross to an olive baboon. So about 3/4 recent olive baboon history, recent olive baboon ancestry. So since from the 70s up into today, we know from both morphological data, which is the basis for this chart, and from microsatellite data, which is what we had up until about 5 or 6 years ago, that the number of Anubis-like animals in Ambocelli has increased fairly quickly, meaning that the proportion of hybrids born in the population has increased and the average hybrid score of individuals born in the population has increased. We've seen fairly rapid scale change in the composition of the population, in other words. This has trait ramifications that we think are interesting. We know that Anubis-like ancestry in both males and females predicts earlier age at maturation, menarche in females, testicular enlargement in males, and a rather large effect on the age at natal dispersal for males, who are the dispersing sex in this species. In addition, we know that more Anubis-like animals tend to obtain higher rates of consortships given the opportunity to consort with a female. And consortships in this species are the context in which most conceptive events happen. So that is a potentially meaningful advantage. We also know that Anubis-like animals tend to exhibit smaller winner than loser effects in ranked competition. That is the outcome of an agonistic encounter. It seems to be less affected by the outcome of previous agonistic encounters. And in work by my graduate student Ariel Fogel published last year, she found that more Anubis-like animals are also more likely to groom with partners of the opposite sex. So a number of outcomes that we think might be relevant to what's going on with these animals. What we're doing now is switching from morphological and microsatellite-based estimates of ancestry to full genome resequencing data, which gives us much more information about the evolutionary history of this population and much more fine-grained information about the types of regions that have been affected by this large-scale change in the types of individuals living in Ambacelli. We've sequenced about 450 full genomes from Ambacelli now, most of them at low coverage, a few of them at high coverage, in work that's led by Ariel, the same graduate student who worked on the opposite sex grooming work, and my former graduate student, Taras Vilgalis, who's now a postdoc at University of Chicago. This is sort of a snapshot of our data. Each line here is a different individual, and they're organized from the most yellow-like individuals in our population to the most Anubis-like individuals in our population on the bottom. And the reason the lines switch colors is because now we're actually able to look across each chromosome and track when they switch from an ancestry that is, for example, homozygous yellow, both copies from yellow ancestry, to a spot in the genome where they carry one allele of Anubis ancestry and one allele of yellow ancestry, to other spots in the genome where both of those alleles are of Anubis ancestry. And of course, they switch back and forth in a pattern that's determined by this sort of complex combination of gene flow rates, recombination in the genome, and natural selection. The whole genome data tells us what the earlier data told us, which is that Ambacelli has been changing over the past few decades, and it's been changing in the direction of becoming increasingly Anubis-like over time. And that change seems to be driven primarily by immigrant males who are systematically more Anubis-like than individuals who are natal to our population, to our study population. One of the things that it tells us, though, that the earlier data did not, is that even at the start of monitoring in our population, when observers considered everyone in this population to be a particular morphotype of a yellow baboon, there was already substantial Anubis ancestry in Ambacelli. That is, even individuals who have no known recent ancestors who were born in an unbroken pedigree before this recent wave of immigration have about a quarter Anubis ancestry in their genome. What that means is our population is now composed of different types of genomes. There are genomes like the one that this female Rongi has. She's a recent descendant of an Anubis-like immigrant, and she has lots of Anubis baboon ancestry in her genome. And then there are individuals like Eclipse, who actually shares a maternal grandmother with Rongi, who have no recent Anubis baboon ancestors, and so have some spots in their genome with a bit of Anubis ancestry, but there's a pretty big qualitative difference between our recent hybrids and what we call our historic hybrids. This is Ambacelli in context. So these are genome segments from about 100, I think this is a subset of 100 individuals in Ambacelli relative to individuals sampled deep into Tanzania or across the Rift Valley in the Masai Mara or into central Kenya. And then this plot here, which has very Anubis-like individuals and very yellow-like individuals here, reflect the Anubis and yellow founders who are wild caught of the Southwest National Primate Research Center colony in Texas. So those are the yellow founders that look very yellow-like and the Anubis founders that look very Anubis-like. So one thing that's really striking to me about putting this into a geographic context is that there's a very, very long border that isn't bounded by, you know, a mountain chain or a big river that precludes movement of these animals across that boundary. And so there's a large boundary that separates yellow and Anubis baboons that seems to be pretty porous. And the hybrids in Ambacelli actually do quite well. In many ways, they seem to thrive. And yet, if you go not very far outside of Ambacelli, you cease to see a lot of these really admixed genomes, which suggests that there are some sorts of factors that must be restricting completely free gene flow. There's something that's acting as a reproductive isolating barrier. We just don't know what it is. Okay, and that just gives you an idea of how long that boundary actually is. Our best guesses are that the responsible factors may be in part behavioral. And that's because we have evidence from a prior analysis of consortship behavior that there appears to be some degree of assortative mating by ancestry in Ambacelli. So these are kind of theoretical patterns that you might see for the plot that I'm showing you in the center, where hotter colors reflect a higher probability of mating for a male and female of a given genetic ancestry, given the opportunity to do so, and lighter brighter or cooler colors reflect a lower probability of a mating event conditional on the ability to do so, that is males and females living in the same social group at the same time and so on. This is really a mix between an advantage accruing to Anubis males in particular, which I told you about earlier, and this pattern of assortativity, which is one way that two distinct diverging taxa can keep themselves from collapsing back into each other, right? If they prefer to mate with other individuals who are genetically similar to them. Similarly, in Ariel's analysis of grooming behavior, what she found is that male and female Anubis-like animals tend to like grooming with each other more than they do individuals who are dissimilar to them from the perspective of ancestry, suggesting again that they're paying attention in some direct or indirect way to the genetic background of their potential social partners. And that's something that can lead to differences in the downstream structure of the genome. Okay. So how might we see this? Well, Ariel reasoned that one way you might see the imprint of assortativity in the genome is that you would start seeing correlations in the ancestry state of animals across distinct chromosomes. So we know that regions of the genome on the same chromosome are co-inherited together because they're physically on the same piece of DNA. But when you're talking about two chromosomes, there's no reason that ancestry states should be correlated across those two chromosomes. So in other words, if we see an individual who's Anubis-like on both copies of one section of chromosome 1 and both copies of chromosome 2, a separate chromosome, that might not be particularly striking. That's going to happen given this admixed structure to our population. But if we see that systematically over and over in our population for those particular loci, that points to potentially regions that are involved in barriers to gene flow, barriers to admixture. So that's what this would look like. Lots of individuals where if we look at the exact same loci, you see this correlation in ancestry states. And you see a depletion of individuals who have opposite ancestry states for that exact same pair of regions of the genome. So this can be caused by hybrid incompatibilities, right? If individuals like this never make it to birth or systematically selected against by the environment, you would get an enrichment of patterns like this and a depletion of mismatch patterns. And that's been the focus of most studies of swordtail fish and mice and Drosophila and so on that have been interested in identifying these kind of patterns. But given what we know about the behavior of animals in Ambocelli, we know that another reason this kind of pattern can arise is if individuals who have opposing genotypes, opposing ancestry genotypes, simply decide not to mate with each other. So we asked about the correlations between ancestry states across ancestry informative markers, that is places in the genome that very clearly differentiate Anubis and yellow baboons across the genome, and identified the very extreme 0.01% percentile of the strongest correlations, the ones that look really unusual in the genome. And we asked, okay, do genotypes at these particular pairs of sites explain mating behavior in our population? So what we were asking is whether, given the potential for a male and female to mate, did they or didn't they, right? And we controlled for the effects of dominance rank, of age, of where females were in their reproductive career, of group demography, and of observer effort as sort of a technical confound. But we are particularly interested in ancestry combinations, right? Can we get any sort of significant signature of mating likelihood based on genotype at a given pair of loci in the genome? Okay, and in that case, we're asking whether individuals who have configurations like this, yellow, yellow, and yellow, 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 and yellow, yellow for both the female and male, or Anubis, 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 Anubis for a pair of individuals were more likely to mate than those who had these sort of configurations of mismatched genotypes. So that's specifically what we're asking. We find 92 locus pairs across the genome that are significant following multiple hypothesis testing correction across the genome. And interestingly, for these regions of the genome specifically, we see a lower proportion of introgress DNA, a lower proportion of Anubis ancestry in the genome relative to the genetic background, right? The genomic background, suggesting that these particular locations are being prevented to some degree from free introgression into Ambocelli. If we look at overall estimates of ancestry that are based specifically on those sites and ask how well they predict mating behavior compared to genome-wide ancestry or a suite of morphological characteristics that we know differentiate yellow and Anubis baboons, we get much stronger evidence that those mating associated sites explain mating behavior, which is a little bit circular, but this is a bit of a sanity check for us too, better than these alternatives. We were actually a little bit disappointed that we weren't getting a clear signature based on morphology, because that might have given us some inkling about what the baboons themselves were paying attention to. It appears from everything we've done to not be any of the things that we pay attention to when we score them morphologically. But if we ask where in the genome these mating associated sites are located, we find that they tend to be systematically enriched for genes that are expressed in neurons and for brain and behavior related phenotypes, which perhaps makes some sense, but is also a little bit of a head scratcher in terms of where to go next. So what we've identified is a relationship between what these animals are doing on a day-to-day basis vis-a-vis one another, right? How they decide to interact with each other in the type of behavioral data that we collect on a regular basis. And a long-term imprint that influences the structure of genetic variation within this population and the level of divergence between these two relatively distinct baboon lineages. What that suggests to us is the importance of linking field observations, which tell us about how, genetic structure arises, with genetic observations, genetic data that give us insight into the history of our population that long predates what we can collect through direct observation. In this case, it was the field observations of new immigration of different looking males that led us to pursue the hybrid question in more detail. Our genetic data told us that there was fairly fast moving change in Ambicelli in recent times, and the field data gave us some idea about why that might be, advantages related to maturation and mating and so on. And the genomic data have been really critical because they give us the signature of events that happened hundreds to thousands of generations ago. And even though Ambicelli is a relatively long-running field study, we will never have hundreds to thousands of generations of data. And so the synergy between those different data types has been very powerful. With respect to human evolution more broadly, I think it highlights the utility of having living primates as a window into things that we simply cannot observe in our own lineages, in archaic hominins, where we'll never have examples of early stage admixed individuals and how they behave. And I think they also sound a note of caution to interpretations of the genetic data alone from Neanderthals and Denisovans that suggest very, very strong phenotypic barriers, or that have led people to infer very strong phenotypic barriers to admixture among those lineages. Because I think based on what we get in Ambicelli, where we actually see very similar signatures, it would have been very hard to predict what those hybrids actually looked like without seeing them. And so with that, I want to thank you all for your attention, and particularly draw attention to the people in my lab who've led this work over time, Amanda Leah, Jordan Anderson, and Rachel Johnston on all the sort of functional genomic work I talked to you about earlier, and Ariel Fogel and Taras Vilgalis for their work on baboon evolutionary history and hybridization. And I'd be happy to take questions if there's time. Thank you.
Thank you so much, Jenny, for that amazing talk. Thank you so much. And we're all clapping here, virtually or otherwise.
Thank you.
Yeah, normally what we do is we continue now just to field questions. And I'd leave it to anyone who has a question for Jenny to please put your hand up. And Jenny, using the Zoom interface, if you're comfortable, you could just take a look at whose hands are raised and field those questions at your own pace. So so we can take it now. I see do you see your participants list? And
I do, and it
okay, great.
Joe Manson has a question. So
okay, yeah, go for
it. Thank you. That was an amazing talk, amazing research program.
Thank you.
My question's about the male dominance and how the higher ranking males seem to age faster. And so you take that there's a reproductive advantage for higher ranking males in terms of like per unit time or production of offspring per unit time. So how much would then the overall advantage of being high ranking be diminished by the fact that they also age faster, if at all?
Right. Okay, I'm guessing here. Okay, so I just want to preface this by saying this is not a calculation. This is what I think the answer is. And I think that the answer from the perspective of fitness, right, just how many genome copies you're leaving behind, I think probably very little. Because I mean, and I think in the baboon males, if you don't play the game, you're never going to win, right? And so they are bearing some degree of physiological costs that we seem to be picking up in the molecular data as well, to the sort of energetic expense of fighting with other males. And I didn't really mention it, but also mate guarding females, you know, pursuing consortships with females where sometimes they forgo opportunities to feed and so on. The only way that they could bear a fitness cost, right, is if you had the comparison between you can maintain that sort of high rank and not pay what may be a physiological cost in terms of, you know, activation of these potentially self-damaging immune processes, and still maintain high rank, right? If you could do that relative to the situation we think we're seeing, which is that males we think can't do that and therefore may be paying a price that eventually leads them to have shorter dominant tenure, right? Because I think that's where the cost would come in that you're asking about, right? Is paying this price going to mean that you're going to be dominant for a shorter period of time? Because we've just established that if you don't pay the cost, you're not going to be dominant at all. And so like there's no advantage to that, even if you live forever, right? So it's only that like if you have individuals who can pay less of a cost, right, then they would have potentially an advantage if this cost leads them to shorten tenure. And the case where that's most likely to happen really are in cases where they don't have much competition, right? Eventually, like occasionally we have males in groups where they are lucky enough to be really the only adult competitive male for some period of time. But that's not exactly the comparison that you're asking about, I think, in general.
Okay, thank you.
You're welcome. I hope that sort of answered the question.
Oh, yeah, very good.
I saw a hand from Dan Fessler.
Hi, Jenny. Katrina, why don't you go ahead? You're middle of the night there, so.
I'm actually in Los Angeles, so.
In that case, no, please go ahead.
Okay, thank you, Dan. Thank you, Jenny. It was really, really interesting talk. So my sort of like, I was thinking if for baboon female, it seems like it's really good to be well social integrated and very high ranking. So then if these females seems like they have a little bit less control of, you know, which rank they're gonna end up with. And there's not such a strong correlation between rank and social integration. Could it be that females sort of can compensate through social interactions with their partners? That there's sort of like, if you know, not everyone can be high ranking, but you can sort of like do it through social integration. And related, if you have a chance, do you think what could be a relationship between rank and social integration? Maybe not in baboon so much, but maybe for macaques, because you could see how the arrow could go both directions, right? And if you have any thoughts on that. Thank you.
Yeah, so I think the really broad answer is it depends a lot on the system that you're studying. I mean, what I've highlighted is a difference in the sort of maintenance and ability to sustain rank just in the two sexes of the same species, that in some senses we mean the same thing when we mean dominance rank, and in some other cases we don't in terms of outcome, right? So so I think I'm going backwards in your question. So in the macaques that we study in captivity, dominance rank is very strongly predictive of the strength of social relationships. But in part, that's because I think we're putting them in groups where they don't know anybody. In a naturally constituted group of Rhesus macaque females, they would also follow this sort of matrilineal inheritance structure. And in that case, in both baboons and Rhesus, a female's closest female social partners tend to be her relatives. Okay, so there's not, you know, a completely unconstrained ability to form strong social connections with other individuals too. If you have a lot of sisters and daughters that is very, very helpful in that regard. So in other words, the best predictors we have of the strength of social connections in females, especially sort of like quantity level measures, is how many female kin do you have in your group as opposed to dominance rank. But there is some ability to potentially compensate by forming strong social bonds with males, which also is a predictor of mortality risk in amboseli. And that's been quite interesting to us to look at, because that looks pretty independent from social connections between females to other females. The constraint there is then you actually do run into rank effects in the baboons. So in other words, male social partners for female baboons appear to be a resource that they are quite interested in developing, and therefore hierarchy plays a role in who gets to develop them. In terms of the interplay between all of this stuff, there's a really interesting paper I can point you to that Susan Alberts led to ask a slightly related question, which is whether female baboons could compensate for early life social adversity by forming close social bonds in adulthood. And the short answer is they probably can't. It's not enough. So there might be a buffering effect to some degree, but it doesn't, we have not seen evidence that it completely offsets the disadvantages of some of these other kinds of things. So thanks. Yeah, Dan?
Hey, so I will first echo Joe's remarks in terms of the amazing nature of the data. And then I'll say this is kind of unfair in the sense that in some ways, maybe Amanda Lay is the right person for this comment, but I'm hoping that I can inspire you and your large network of collaborators to use some of the approaches that you have in thinking about some broader questions. So thinking about the gene expression work, you know, there is, I wouldn't say there's a debate in the literature, because that's probably, the issue hasn't received that much attention to call it a debate, but there is discussion as to whether prestige in humans is an entirely derived set of, you know, behaviors and social orientations and corresponding cognition and affect, right? Or whether it's really just a modification of an ancestral dominance system, right? And sort of psychologically, what that boils down to is, you know, are successful high prestige people actively suppressing dominant strategies, or do they not have to because they just, prestige is what appeals to them, right? So it strikes me that that the gene expression work potentially offers a really valuable tool here, because if it's the case that you know, that winning at the national spelling bee or whatever it is, right? Whatever the high stakes competition, right? And I'm intentionally leaving athletics out of it because athletics are arguably sort of proxy dominance contests, right? But winning in a pure prestige competition, winning an election maybe, or something like that, right? If individuals who succeed with those strategies look similar to individuals who succeed with dominant strategies, right? Paralleling the baboon male-female, or if they look different, right? Rather paralleling the baboons, right? If they look very different, they have different strategies, right? Then that tells us, I mean, you know, we can look into the past in a way that there's no way we could otherwise, right? And unlike the baboons and the macaques, people can tell us what their strategies are, right? That they can describe their social orientation and their standing and how they address contests, right?
Yeah. I mean, are you asking whether you could use these types of outcome measures as a common currency to compare? Yeah, I think that would be fascinating, actually. I mean, I don't really study humans, but humans are remarkable in our flexibility and creativity and coming up with different sort of status systems. One of your colleagues at UCLA, you know, Steve Cole, has done quite a bit of work on sort of gene expression signatures of social stress and some social isolation kind of work. And in general, his take has been there's a remarkably conserved signature, where I think by conserved, he might be using it in a bit of a different way than an evolutionary biologist would use it. But there's a remarkably consistent signature across the sort of studies that he's done. Now, I don't think the studies that he's done have, to my knowledge, encompassed the sort of variation you're asking about. And I think it would be fascinating and very, I should say, very doable. I mean, in principle, very doable to collect those kinds of samples in different kinds of populations, where folks who know those populations well can think about the right way to objectively or subjectively measure prestige, status, dominance, and so on. I think, like, for me, when I think about comparative work to try and test the hypothesis that I sort of presented to you, that it's really about, like, are we being nepotistic here, right? And it's not about individual characteristics, or are we, you know, is it all about individual strength and condition? I tend to think about larger, what may be larger contrasts, which are differences between species. You know, is it energetically expensive to be a dominant meerkat? Is it energetically expensive to be you know, a dominant capuchin, or so on? But I mean, yes, I like within humans would be amazing. It would just take a lot of work to collect samples.
Right, but well, first of all, I should say between population comparisons in humans are certainly possible, but they're not necessary, in the sense that you know, no large society is homogeneous with regard to the strategies that individuals pursue. There's a lot of variation within that. I mean, it seems to me that it's an empirical question that is difficult to answer, whether just with standard things like, you know, C-reactive protein or whatever, right? Whether you know, achieving high status through prestige and achieving it through dominance have similar outcomes, are very different ones. And this would potentially give us an opportunity to really answer the question of the derivation of prestige. I don't, like, it's the first time I've ever thought of a way of answering that question.
Yeah, so I think it would answer one aspect of your question better than the other, which is, what are the consequences? What are the consequences I feel pretty comfortable with? What the roots are, I'm not sure about, because you can imagine that, you know, it sucks for whatever reason to be low prestige, and it sucks for whatever reason to be, you know, low on a hierarchy that's determined by physical dominance. And both of those things kind of stress you out, right? And then they could converge on a similar biomarker signature without necessarily having the same, you know, origins. But if you want to ask a question about, are the consequences very similar, then I think that there's something there's a road that could be gone down. I mean, one of the things that always seems to me very difficult about humans, though, is just, you know, you take a single human in a society like the ones that we live in, and, you know, people are high status at work, but low status at church, and, you know, their softball team is awesome, but their, you know, relationship within their family is not. And so you have so many overlapping sources of measuring hierarchy that they seem difficult to disentangle. You know, whereas we study non-human primates in part as a model because, like, relative to us, it's quite unidimensional.
No, those are all good points. I see Kim is waiting, so I'll be quiet now, but fascinating. I could talk to you for hours.
Thanks. Hi, Kim.
Hi, thanks so much for this talk. You guys are doing some really, like, exciting projects, and it's cool to hear about them. Yeah, I was just wondering if you guys, like, have anything going on looking at gene expression in kind of like a longitudinal way. So, like, following and, like, resampling males and females, like, as they develop, to see, like, when these gene expression patterns become set and how they, like, develop, like, if it's like a sudden jump for, like, some set of genes, or if it's, you know, more gradual. And then if you're, like, with that, if you could do, like, predictive modeling with males to, like, see, I don't know, maybe not. It's probably like a response, actually.
I mean, that's really, like, I wish we could. Here's what I'll say is that we do some longitudinal sampling. Our longitudinal sampling is constrained because sometimes animals that we're studying do things that are inconvenient, like disperse or die. And then we can't sample them again. And while we're pretty good at targeting animals for darting. It also just depends a lot on the conditions when we're out there and who we can get. So we are sort of, we slowly aggregate cross-sectional samples, and we even more slowly aggregate longitudinal samples. We do find the longitudinal samples extremely useful, right? Because even when they're a subset of the cross-sectional samples, because like, for example, in the epigenetic clock stuff, we have a subset of males who we've sampled when they were at different ranks. And then so we could just ask, well, do you look like where you are now? Or do you look like where you used to be? And the answer tends to be you look like where you are now. That is, males who actually fall in rank, they increase in chronological age, but relative to their chronological age, they start to look, epigenetically younger. So that's really useful to see. We also have repeated samples for some individuals for rank, but we don't sample really young individuals, so we don't have an ontogenetic trajectory, which is, I think, partly what you would be asking about. And we probably don't have enough of the right sort of samples to develop a good predictive model. Nevertheless, we do have some evidence that suggests that those particular molecular physiological states that we're measuring are not just a consequence of dominance rank, but that they actually precede those individuals who attain high rank. So your question is a great one. I can just imagine what my students would say about the samples that we have. It's like, what about doing this? I think we're not there yet, but maybe in another 50 years or so.
That's really cool. Thank you so much.
Yeah, that's a great question. Hi, Abby.
Hi, Jenny. Thanks for spending your part of your day with us. I enjoyed your talk very much. I'm going to lower my hand. So my question dovetails nicely after Kim's, but it's a little bit more specific in the expression data stuff. I was curious if you, in the analyses where you use male data to predict female rank and use female data to predict male rank, what happens when you mask the immune genes? Are the remaining, like if you mask, because it's such a driver of the female signal, I'm just wondering if then perhaps there's some overlap and some similarity that might be indicating some biological pathway that's shared.
Yeah, what an interesting question. We have not done that. It'd be easy to do. My guess is that if we did it, we would lose a lot of predictive power just period, right? Like our prediction accuracy would kind of go in the toilet. But if it didn't, then I think you're right that it has potential to pick up some features and genes that maybe have some shared effects. We focus a lot on the immune pathways that we pick up, and in part because they're strong predictors and they come up a lot in our analyses, and in part because we feel like we understand them a little bit better, but we often pick up metabolism related stuff and mix signaling, which does everything, right? And so there are these other pathways that I think we have not paid nearly as much attention to, that might speak to your question.
Yeah. Cool. Thanks.
Brian?
Yes, Jenny. So I was wondering if there is any insight that you could add to this kind of larger order species level pattern of increased. Is it an increased range of olive baboons at the expense of yellows? More is that it? Actually, that's my first question. Is like what is that larger order pattern? Is it, and if it's yellows winning ground against olives, or vice versa in general, but also just to, regardless of what that particular pattern is, I was wondering if there are things that you could use from your research, both at a behavioral level, demographic level, and genetic level, to shed some light on, is this a story of one species having a greater likelihood of successfully dispersing to a neighboring population of unlike individuals? Or is this, for example, a story about dispersal plus some kind of advantage, conditional on being a male in a group that is dissimilar, do these dissimilar males have any mating advantage, or is it, basically a coin toss whether or not, a dissimilar male is going to be overall more likely to reproduce in a group in which he's dispersed into, or just any relationship between in any light that you can shed on those like higher order processes from your field site.
Well, so the hypothesis that olive baboons are expanding at the expense of yellow baboons and other species is something that Cliff Jolly has talked about for some time. He thinks that may be true. I think we don't know strictly whether that's true. It's possible, but what I can say so far to inform that question is the location of the hybrid zone in Kenya seems to have been relatively stable for really what in an evolutionary sense is a short blink of a time, which is since the 60s when people wrote about it, first outside Amboseli. So at least within that period of time, there's no obvious big shifts to my knowledge. The second thing is that we've done some modeling of the genomic data to ask about what the nature of the contact between Anubis and yellow baboons has been in East Africa. In other words, did they diverge? And what we're seeing is just the first recontact, right? That's a very simple model. Or did they diverge? And there's been like kind of this steady low background gene flow since they diverged, or something else. And while I can't tell you what the something else is, our data suggests it's something else, right? The patterns of the genome sort of it's kind of the pattern of blocks of ancestry tracks of the genome are not consistent with a single divergence and then recontact event. In other words, it looks like they've been doing this periodically for some time, for some reason, but not in a continuous manner. And that's consistent with the fact that like in Amboseli, you can see all these historical hybrids, and then you can see these recent hybrids, and it creates this kind of bimodal pattern that I didn't show you of ancestry, suggesting that there have been a couple periods in which they kind of exchanged a lot of genes, and then they maybe stopped for a while for some reason, and then they did it again. And I think that leads back to the fundamental question of what's keeping these things apart. I mean, primates in general hybridize a lot in nature. And, one explanation is that primatologists love to split. And so they're not really species anyway, and they're not, it's not hybridization, it's just, separate populations. But I think just to attribute it all to that is kind of not very interesting. There clearly is hybridization even between fairly diverged lineages, and what's keeping these things apart as opposed to them collapsing into one another is a big question that we don't know very much about, where other people who study non-primates are way ahead of us on that front. So that periodic, that what we think is a periodic recontact would also suggest to me that there's not a simple, Anubis baboon advantage over other primates. In terms of mating advantage, Anubis-like males have some mating advantage in Amboseli. But we don't know whether that's because they are Anubis males in a yellow baboon zone, or whether it's because they are different.
Yeah. Okay. And it does that, so that mating advantage is something that you can track from your demography, of course, coupled with your genomic, more detailed genomic data. Is there anything behaviorally that you see as corresponding to, the how question of how does that mating advantage sort of transpire at a behavioral level? Is there anything that you care to say about like the game, that they're playing and how, for just to be able to try to narrow down some of the genes that would be more likely to introgress, maybe that would be useful information.
I wish I knew the answer to that. I think I think is a question we have data to look into much more deeply. So what we have right now is you're exactly right. It's just based on the mating records and the genetic data. We know that we see a slight advantage to Anubis-like males, but that's controlling for things like dominance, rank, and group demography and stuff like that. I mean, the biggest thing is rank, right? I think if you're a high-ranking male, then you're going to mate with whatever of the females are available to you in that group during your period of high rank tenure. But I think your question gets to, well, are they better at forming coalitions? Are they like, do females, you know, like them better? What are they like, you're asking about a behavioral mechanism. Yeah, I don't know the answer to that yet, but I'd love one of our students to want to ask that question. So
yeah, well, great. Thanks a lot. It's wonderful that you're permitting, you know, all of these relationships to be explored, and this work between behavioral and genetic measures of these processes is just fascinating. So thanks.
well, thank you to all of you. I know it's hard to sit on the computer for one more hour during the day, so I really appreciate it. And I hope that I get the chance to actually have long, open-ended, in-person conversations with many of you in the future when it becomes possible.
Yeah, it's been a great honor for you to join us, and this is a great way for us to start our week, to spend some time with you and to hear about your work. So we just want to all thank you again for all of this, and of course also for being willing to meet with students. You might get a late breaking email from a student or two who would also be curious about your schedule, just to warn you about that. But you've been so generous with your time, and we really appreciate you bringing your work to us.